On this set of slides, we will review over the histology of the reproductive system. We will begin with the female slides and then change over to the male slides. Here we have a slide of a primary oocyte. This is for a female, and we call the eggs the oocytes. So here we have, so we can get this pointer to working, a primary oocyte. Now we know it's primary because we have one cell layer of follicular cells. So one cell layer is primary oocyte. Now here we have a secondary oocyte, and we know that because we have two or more layers surrounding the oocyte. Now here we have a mature follicle or a Graafian follicle. We can see the oocyte here with our zona pellucida cells, and then we can see our granulosa cells. This large fluid-filled area is the antrum, and this oocyte is just about to be ovulated. So when we look at the female slides, we look at the primary oocyte with one cell layer, the secondary oocyte with two or more cell layers, and then our mature follicle with our oocyte and our large antrum. The remaining slides will be of the male reproductive histology. We are beginning with the testes, and we are looking at the seminiferous tubules, and that's each of these represented by the ST. The open area is the lumen. The clusters of cells to the outside are the interstitial cells of Leydig. These produce the testosterone. So the seminiferous tubules for sperm development, the lumen, and the interstitial cells of Leydig. Here we have the seminiferous tubule slide, and with that we have the interstitial cells of Leydig that produce the testosterone. We have our seminiferous tubule. The outer part is called the basement membrane. The centers are lumen. And when we look at the stages of development of a sperm, there are 5 stages of development as they move from the basement membrane to the lumen. We will identify 3 based on location. The first that we have is right up against the basement membrane. These dark circular dots are going to be the spermatogonium, or the first stage of development. As we move out and we get midway out, we will have our spermatotids, and then as we get to the interior part where we see the tails of the sperm, we call those the spermatozoa. This is a close-up of the previous slide where we can see our basement membrane, the entire structure being the seminiferous tubules. We see our dark dots as our spermatogonium. Moving towards the center, we will have our spermatotids. And then we have the spermatogonium, and these are what we think of when we think of sperm, with the flagella. And there are other stages. There's the primary spermatocyte, and there are the Sertoli cells, etc. However, we will just identify the spermatogonium, the spermatotids, and the spermatozoa. This slide is of the Ductus epididymis, and on this slide we can see all of the connective tissue. We can see that we have the spermatozoa, or the sperm cells. We have the pseudostratified columnar epithelium right in this area here. We're looking at the ductus deferens. You may see some smooth muscle in with the connective tissue. This is a close-up of the ductus epididymis. Again, the lumen. You may see some of the spermatozoa, the pseudostratified columnar epithelium, the connective tissue with some smooth muscle.